Okay, this lecture is about microscopy. This semester, we'll talk about lots of different ways and techniques to study cells. First one we're going to talk about is microscopy. This is used to look at cell structure, look at gene expression, and it can be very, very specific. So we're going to talk about four different types of microscopy. We're going to talk about light, fluorescence, and scanning and transmission electron microscopy. You don't have to know the details about how each microscope works, but I want you to understand how they're similar and different, and most importantly, what you can and cannot see. So light microscopy, oops, is the type of microscopy you have used in school. So it uses light to illuminate the specimen, and that's what we can see. Light microscopy has a magnification usually up to about 1,000-fold. So you can make something 1,000 times bigger than it is to take a image of it or to look at it. The resolution is to 200 nanometers. Resolution means can you distinguish two different things under the microscope? So if this um, space between is greater than or equal to about 200 nanometers, you can see it. If um, two bacterial cells are closer than 200 nanometers, you cannot resolve it. So it would look like more like this. Like you couldn't see that they are two distinct spots. Okay. So light microscopy has really the kind of the lowest magnification. We'll talk about electron, which is much higher magnification, and lower resolution. But you can see live and dead cells. Dead cells are usually stained, and we stain them because if you see here, this little guy, this paramecium, is really see-through. And so to get more details, we use staining, but staining kills the cells. You can see both prokaryotes and eukaryotes. And I'm going to show you a slide in just a second about some of the organelles you can see. But again, most cells are see-through unless they have chloroplast. And this is really the only color, natural color you can see with a light microscope. And these, in case you're wondering, these are my favorite little guys. These are called euglena, and they're a protist, and they have a flagella, and they have chloroplasts, so they're green looking. And you can see that color. The other color you can see is red blood cells. So I encourage you to look at these two videos and just kind of see what you can see with a light microscope. Now we can get a bit better resolution by playing with how the light refracts. So this is phase contrast, and this is interference contrast. Um, but we're not going to distinguish those. So these are all considered by me just light microscopy. You don't have to distinguish the type of light microscopy. You can see they're all in gray when it's live. Again, fixed samples down here show that they're usually stained so that you can see the details better. You can distinguish different cells. And this slide is a great reference slide for you. So what can you actually see with my light microscopy? So you can definitely see eukaryotic cells. And in those new eukaryotic cells, you can see a nucleus in sometimes, not always. You can imagine the cell membrane. You can see chloroplasts. If it's a plant cell, this is a plant cell or a protist. You can imagine the cell wall of that plant cell, though you can't distinguish the cell wall from the cell membrane. And of course, you can see the cytosol, all that fluid. Sometimes, if we stain the cells, you can actually see chromosomes. Change color here really quick. So just to orient you, there's a cell. This is a plant cell, so it has a cell membrane, cell wall. The dark big blob, that's the nucleus. And because we're staining, you can also see the nucleolus. So that's dark staining. And then chromosomes, this is a plant cell undergoing cell division. You cannot see mitochondria or the ER or the Golgi. And yes, excuse me, in this amoeba, here's the nucleus. We have lots of these little vesicles, organelles, who knows, but you can't distinguish them. So we're really limited to the nucleus because it's so big, the cell membrane, cell wall because it's around the outside, chloroplast because they have natural green coloring, and chromosomes when we're staining and a cell's undergoing cell division. I want you to note here, like in this cell here, you cannot see chromosomes because they haven't condensed, they haven't gotten fat, they're not visible under light microscopy. Second type of microscopy, which is my favorite, is fluorescence. Fluorescence means that we have a ultraviolet light, UV activated molecule that under a UV light, we see color. Okay. So again, this is a type of light microscopy, but we're always going to refer to it as fluorescence. What you're going to notice, oh, and these are always dead. Because to get the fluorophores in there, or I'm going to tell you about antibodies in a minute, you have to kill the cells so you can get these big molecules inside. So fluorescent probes is what I'm talking about, or a tag. Fluorescent microscopy is always going to have a did it again, black background and bright colors. Okay. You can see much more detail. It has about the same resolution and magnification as regular light microscopy. You just can see details because of that staining, because of that coloring. Fluorescence just means we have some kind of, as you can see here, fluorescent dye, some kind of fluorescent molecule that's very specific. Or we will also talk about immunofluorescence. And that's where we use an antibody plus a fluorescent dye to stain the cells, stain the molecules, identify the molecules. So just a heads up, this semester, anytime you see immuno, that word, it means we're using an antibody. Excuse me, trying to get a drink. Okay, so look for that black background and bright, vibrant colors for fluorescence. There's another kind of fluorescence that you can use for live cells, and that's called GFP. And GFP stands for green fluorescent protein. GFP. And when we use GFP, we're manipulating the cell genetically. Requires genetic manipulation of the cell. And what I mean is we make something called a GFP fusion protein. So at the DNA level, we take the gene for GFP and we connect it to your favorite gene. And so it's kind of showed here when this protein that you're interested is expressed, it will glow green because it has this fluorescent molecule literally attached to it through the protein structure. GFP was found in Aquaria Victoria, a jellyfish that naturally fluoresces. And then we took advantage of this fluorescence and have sequenced it and figured out how to, like I said, attach it, fuse it to another protein. The beauty is, like I said, you can see live cells because you're not having to get any fluorescent dye or antibodies into that cell. So it's a really powerful technique. So in this case, you'll see bright green on a black background. And what I will do is distinguish if something is a live cell or it's had genetic manipulation or you've created a fusion protein. And that's how you'll know this is GFP and not some kind of fluorescence. Okay. Third type is transmission electron microscopy, also called TEM, and it uses electrons to create the image. TEM, again, stands for transmission, and it gives us this beautiful 2D image. Excuse me. So I want you to look at this image. It looks flat, but it has lots of details. Okay, I need to stop writing above that line. Right? This is actually the nucleus and the ER with ribosomes. You cannot see this at all with light microscopy. And you could not see this fine of detail even with fluorescence. So the magnification, I've seen all kinds of crazy numbers, but we're just going to keep it as greater than 1000x. The resolution can get down to about 1 nanometer. So if two dots are even just a nanometer apart, you can see the difference. So you can actually see all these individual ribosomes, right? Here and here and here and here, even though they're very close together. So higher resolution, higher magnification, lots of details, and the image is nice and flat, 2D. In contrast, scanning electron microscopy, that's where we get, and this is SEM, that's where we get 3D images. And I love these. So here, stereocilia are in your inner ear and help you hear. Here it is under light microscopy, and here is that same structure under SEM. So I want you to see the difference of this 3D, and you can see it looks like little balls all around it versus a 2D image with transmission electron microscopy. They both have about the same um, magnification and resolution. Resolution on scanning is a little bit less. Oh, I can't spell that word today. Resolution, it says here between 3 to 20 nanometers. Um, and that's just because of that 3D effort. But again, we're using electrons to create the image. Right? So looking side by side, anything that's looking nice and flat um, is transmission. And yes, I know this DNA molecule looks 3D, but it's still transmission, just letting you know. Over here, these are red blood cells and a white blood cell, and these are macrophages. And again, you can imagine this 3D, you can see depth in both of these images. Now, transmission and scanning are always going to be in grayscale. There's no color that naturally comes. But you could do something called false coloring, where the computer will add color to certain objects, certain cells, to just help distinguish a little more. Let's look together at two eukaryotic animal cells. Both are in grayscale, but I want you to notice the difference in detail. Okay, so this on the left is light, and on the right is transmission. So you will be asked to determine what kind of microscopy different images were generated with. So again, take a look at the amount of detail. Do things look flat? Do they look 3D? Or are they on a black background with bright colors? All right, that is our chapter 1, end of chapter 1. And again, you have learning objectives, but here are the general things we've covered: parts of the cell, microscopy, and the theory of endosymbiosis. All right, thank you all, and I will stop.